There, my name is Courtney Richardson, and I'm going to be doing a presentation on signal transduction. So let me share my screen, and we can get started. Okay. Signal transduction is the process in which a cell responds to an outside signal involving the movement of information from the outside of the cell to the inside of the cell, where the reaction occurs within the cell. The external stimulus could be in the form of energy or molecular change, such as the binding of a signaling molecule, also known as a ligand, to the cell surface. Once a signaling molecule or ligand has binded to the receptor, the signal is amplified and it activates a molecular pathway. It's also referred to as, things happening downstream, and the changes within the cell are able to begin taking place. In some cases, signaling molecules are able to cross into the cell through the cell membrane. And an example of a signaling molecule, just in general, would be a neurotransmitter, maybe a hormone, maybe a pheromone, things like that. So let's go on to our next slide. So now we're going to go into the receptor tyrosine kinase mechanism. Receptor tyrosine kinases are a group of receptors that are located within the cell membrane. They act as signal transducers that mediate cell communication with other cells. Cellular processes such as apoptosis, differentiation, proliferation, and metabolism are just some of the functions that tyrosine kinases are involved with regulating. So they are involved with many big things that happen within the cell. So let's go on to normal tyrosine kinase activation. There are 90 known tyrosine kinases, which are then divided into receptor tyrosine kinases and non-receptor tyrosine kinases. So out of the 90, we have 58 that are receptor tyrosine kinases, which are further then divided into 20 subfamilies. And then we have 32 non-receptor tyrosine kinases divided into 10 subfamilies. Receptor tyrosine kinases have similar base structures with an amino acid terminal, an extracellular domain, an intracellular tyrosine kinase domain, and then a tyrosine-rich carboxy terminal, as well as a juxtamembrane region. The activation of receptor tyrosine kinase is initiated through the binding of that signaling molecule, that ligand, to the ligand binding site and receptor dimerization. Once the dimerization occurs, an intracellular tyrosine kinase domain is activated through the transmembrane domain. Transphosphorylation of the tyrosine residues in the activation loop, juxtamembrane region, and C-terminal sequence are required for activation. Phosphorylated tyrosines become binding sites for signaling molecules, also known as ligands, which then bind to the phosphotyrosine residues. So some of the proteins that dock into this phosphorylated receptor tyrosine kinase activates further signaling pathways that regulate then processes such as those big things that we mentioned earlier: cell survival, proliferation, differentiation, metabolism, and as well as the overall control of the cell cycle, as well as apoptosis or programmed cell death. So let's go on to some abnormal tyrosine kinase activity. Abnormal activation of the receptor tyrosine kinase involves the receptors, the partner molecules, and the surrounding environments. There are 4 proposed mechanisms that lead to an inappropriate activation, as you can see here. For example, increased kinase activity through overexpression, gain of function mutation, chromosomal translocation, as well as autocrine activation, all leading to this increase in kinase activity. Aside from these 4 main mechanisms of atypical activation, oncogenic receptor tyrosine kinase activation can also be influenced through kinase domain duplications, microRNAs, tumor microenvironmental changes, negative receptor signaling regulators, as well as altered endocytic genes, and spatial deregulation of receptor tyrosine kinases. So let's kind of go on to how cancer is involved in the tyrosine kinase receptor. So oncogenic signaling by tyrosine kinase receptors are known to be related to the formation and progression of cancerous masses or tumors. Let's continue on to our next slide. So here we see the general principles for the receptor tyrosine kinase. You can see that's abbreviated here as RTK, which is very common to see the receptor tyrosine kinase abbreviated like that. Co-activation, let's get into that because this is what this slide is all about. So new research has shown that tyrosine kinases act typically in cooperation as a network of multiple receptors that collaborate, communicate with each other. This is a concept known as receptor tyrosine kinase co-activation. So this means that instead of viewing these receptors as a singular unit, we view them as a network of units, all working in collaboration in tandem with each other. So this complex network working as a group makes it a little bit more complicated to kind of pinpoint the individual because they all work together. But anyways, receptor tyrosine kinase co-activation is commonly found in multiple cancer types. This includes lung cancer, bone cancer, soft tissue cancers, glioma, breast cancer, ovarian cancer, mesothelioma, excuse me, and more, and also more types of cancers, various cancers. So the cancers that are associated with receptor tyrosine kinases, they range from genetically simple tumors, which would originate from a single driving force, to cancers that are far more complex, things that we're going to find kind of above the shoulders, head and neck cancers. So let's move on to our next slide. So let's kind of go into these mechanisms of receptor tyrosine kinase co-activation and how that contributes to the cancer growth and development. So it is theorized that cancers may have kind of evolved this receptor tyrosine kinase co-activation as a driving force to increase the tumor growth and limit the effects of microenvironmental therapeutic attacks to try to control these tumors from growing out of control, basically. A functional analysis of the kinase inhibitors and RNA interfaces have shown that the activation of the receptor does not always mean an attraction for the kinase molecule. And that is why scientists and people that study this have found it very challenging to work toward a theory which the multiple receptor tyrosine kinases are functionally related to. The coactivation networks of tyrosine kinases, they're highly dynamic. They are adapting rapidly. So the measures of direct kinase phosphorylation and direct functional effects within the coactivation networks are very difficult to correlate. Distinct receptor tyrosine kinases are able to maintain their specificity and diversity within the activation of the downstream signaling events, which can be seen through their differential intensity, their localization of signaling, their signaling in response to epidermal growth factor, nerve growth factor, or the kinetics of signaling. So let's move on now into the variations in tumor types and cell lines. So the receptor tyrosine kinase coactivation is commonly found to be the mechanism that resists the tyrosine kinase targeted therapeutics that we had spoken about before. It would benefit the treatment of tumors to understand this kind of relation of gene expressions in patterns of different types of receptor tyrosine kinases within the cancer cell line encyclopedia. For the co-expressed mesenchymal epithelial transition factor, fibroblast growth factor receptor number 1, and epidermal growth factor receptor, there were a large set of cell lines that kind of co-express these factors. So let's move on now to how are we going to treat this in cancer therapy. So the alteration of receptor tyrosine kinases can occur in many different cancers. These findings have led to tyrosine kinase inhibitors and therapeutic agents to try to treat cancer. One commonly prescribed tyrosine kinase inhibitor is the epidural growth factor receptor, which is used against non-small cell lung cancer, for example. So we have first, second, and third generation epidermal growth factor receptor tyrosine kinase inhibitors that, have been formulated with different mechanisms used to try to target specific activating mutations. The use of tyrosine kinase inhibitors are eventually, unfortunately, resisted, although initially they can be very effective. During the treatment of tumors using tyrosine kinase inhibitors, tumors typically either from the start, they start to respond really well before they kind of start to resist it, or they just do not respond at all, which is kind of what they have found with these types of drugs. So the ones that do respond very well kind of straight off of the bat over time will lose their efficacy, which we will go into a little bit further on how we can kind of combat that. So drug resistance. The resistance to drugs is known to occur when treating cancer cells. So typical cancer treatments, typically they want to induce our programmed cell death, our apoptosis, which causes the destruction of the cancerous mass by destroying all of the cells that make it up. So concentration of the cell membrane transporter within the cancer cell may be increased within the course of treating the tumor. So both tyrosine kinases and non-receptor tyrosine kinases are overexpressed within tumoral growths, which then stimulate the spread of the growth. In the majority of tumors, the epidural growth factor receptor tyrosine kinase is overexpressed, which is then why our tumors grow so rapidly, which also results in the autophosphorylation, which triggers the signal transduction of pathways leading to that tumor growth and that spread. So this is a medication. It is definitive. Also known as ERISA, has been proven very beneficial within certain cancer treatments by inhibiting an active non-receptor tyrosine kinase. So patients that have non-small cell lung cancer, whose tumors also contain a mutation of the extracellular growth factor receptor can benefit from this type of treatment with definitive as an alternate to chemotherapy. And they've also found that quality of life increases with a medication that works in this way as opposed to chemotherapy. Other tyrosine kinase inhibitors have proven to be beneficial in cancer treatment. Some inhibitors are, for example, competitive inhibitors. They compete with that kinase for the ATP. Others prevent the interaction of chaperone proteins and tyrosine kinases, which eventually degrades the enzyme. Receptor tyrosine kinase mediates the emergence of resistance to inhibitors through their tumor development alterations, such as overexpression and mutations, which lowers the efficacy against the tyrosine kinase inhibitors in cancer treatment. So this challenge with cancer treatment has provided researchers an opportunity to explore alternative options to these inhibitors because the body just becomes resistant to them, or not the body, but the cancer itself is what I meant to say. So as researchers have kind of branched out, they have found that naturally derived substances in comparison with cytostatic pharmaceuticals minimized the possibility of, for example, drug interaction or side effects. So let's look at some of the kind of more naturally derived plant species compounds that may be beneficial in treating this. So the first one that we're going to look at is curcumin. It is a hydrophobic polyphenol that is extracted from the herb Curcumin longa, which is also known as turmeric. Various studies have shown that there are many benefits with this substance. It is anti-inflammatory. It is an antioxidant. It is antimicrobial. It is anti-metastatic. It has anti-angiogenic properties, all leading to the use of curcumin in cancer treatment, as well as many other medicinal purposes. So research based on the therapeutic strategies of the use of curcumin have shown very promising data in the use of this as a therapeutic cancer agent. Curcumin molecularly targets transcription factors, inflammatory cytokines, protein kinases, microRNAs, and the tumor suppressor gene. So within the realm of cancer treatments, the role of curcumin involves its ability to inhibit the cell proliferation and induce apoptosis. The components that regulates processes like these are receptor tyrosine kinases, and curcumin targets tyrosine kinase receptors, which downregulates the receptor tyrosine kinase signaling pathways. Additionally, when introduced in combination with tyrosine kinase inhibitors, curcumin has proven to help the body overcome resistance with the cancer treatment. So very promising. The hydrophobic nature of curcumin limits its bioavailability, however, which leads current research to kind of study potential modifications that they could make to the substance to therapeutic parameters, which could maybe allow for better bioavailability, better targeting of the tumors for the use of this substance. And our next substance that is kind of new as far as research goes in this new kind of role of seeking out more natural methods to reduce potential drug interaction is you'll have to excuse me if I'm pronouncing this wrong, but it's diosgenin. It is a steroid that is actually extracted from the rhizome of the Dioscorea plant, and it is one of the major bioactive compounds found in the fenugreek seed. Traditionally, this is used in a lot of Chinese medicine. This Dioscorea plants are used to treat everything from digestion, circulation, just reducing inflammation in general. However, recent studies have shown that the use of this substance has a number of other pharmaceutical benefits, which also include blocking tumor progression and increasing the survival rates of patients with cancer. very promising. So deosinogen has a structure that is actually very similar to estrogen. And studies have reported that the use of deosinogen has shown a reverse in multi-drug resistance to cancer cells as well. So very promising. So deosinogen is it has been known to, inhibit the metastasis of cancers such as prostate cancer, gastric cancer, lung cancer, breast cancer, liver cancer, renal and colon cancers, as well as melanoma even. It inhibits these cancers through suppressing the pro-inflammatory and pro-signaling pathway. Additionally, it also sensitizes cancer cells to standard chemotherapy. Again, kind of combating that drug resistance that we see a lot. Excuse me. Some factors that enable tumors to sustain growth, supply essential nutrients, are growth factors, the ability to resist chemotherapy drugs and develop resistance. So it has been found to kind of inhibit the invasion and migration of breast cancer cells, as well as suppress the polymerization of actin. So cortical actin binding protein, or coractin, this is a protein that is located within the cytoplasm, and it promotes polymerization and rearrangement of the actin cytoskeleton. Coractin and actin polymerization. It is directly involved in the formation of cortical microfilament actin cell skeleton, which is also one of the main substrates of the intracellular tyrosine kinases. The tyrosine phosphorylation of coractin changes the structure, which is an essential portion of the cortical microfilament actin formation. Coractin phosphorylation through the kinase is necessary for invadopodia formation, which is mediated by coractin. Tyrosine kinases promote the cell migration through the phosphorylation process of coractin. So now let's go on to invadopodia. The formation of invadopodia, a type of membrane structure, is essential to maintaining the migration of cancer cells to new areas of the body. Invadopodia, they're rich in filaments made of actin and cortical actin binding proteins. Invadopodia membrane structures are formed in the early invasion and metastasis stages of tumor cells. The structures are formed within 3 stages. So the formation of the core is stage 1. The stabilization of the precursor is stage number 2, and then the whole maturation of the invadopodia is stage number 3. Once the core is formed, which may only take a few seconds, and the tyrosine kinase substrate rapidly binds the core and kind of mediates that binding of the precursor complex, which then stabilizes it. So in conclusion, in various types of cancer, the membrane phospholipid specific phosphate gene has been mutated, which results in a continuing signal for cell division and growth, leaving us with those tumors. Growth factor receptor that kind of initiates through the tyrosine kinases include epidermal growth factor and vascular epidermal growth factor, and the involvement of tyrosine kinases malfunction in cancer continues to be studied for potential new therapeutic methods as we all try to kind of push towards finding an ultimate cure for cancer. So that is going to conclude my presentation. Thank you for listening, and I hope you learned something.